Just to introduce our speaker briefly, Dr. Stephen McCarroll is the Dorothy and Milton Flyer Professor of, make sure I get it right, Biomedical Science and Genetics here at Harvard Medical School. He is also a member of the Broad Institute, where he directs the, again, to make sure I get it right, the Genomic Neurobiology section within the Stanley Center for Psychiatric Research. Steve did his PhD with Corey Bergman at UCSF, and he then made the big leap from both in terms of species, from nematodes to humans, and geographically from California to here in Massachusetts, when he came to the Broad Institute to do a postdoc with David Altshuler studying human genetic disease. He joined the Harvard faculty, I believe, in 2009, and has been here ever since. I first heard about Steve's work in 2015. I remember vividly he published this absolutely spectacular paper in which he introduced droplet-based RNA, single-cell RNA sequencing, and they had this terrific story in which, if I'm remembering right, in a single study, they were able to rediscover almost every cell within the mouse retina that had been described in the previous 100 years of research, all in one paper. So that sort of blew me away. I think many of you know about Steve's more recent work on complement and schizophrenia. Again, received a great deal of attention, and absolutely fascinating story. He went on earlier this year, as Kate mentioned, to publish another twist to that story, connecting schizophrenia to autoimmune disease. And in fact, within a day of that paper coming out, I got, I think, at least 2, maybe 3 requests from people to invite Steve as a speaker to the Women's Brain Initiative because of the link to sex differences in these different conditions. So we're delighted that he was able to accept, and that he's able to be here today. And with that, I will turn it over to Steve. Thank you.
Oh, well, thanks for inviting me to come and share this work. Yeah, I was excited to be invited to visit with this group that's been thinking about sex differences and sex-biased biology, especially in the brain, which I think is just a fascinating topic. It was actually something that we were interested in for a long time, but didn't actually didn't really know what was a great way to study. And in this case, actually didn't, in the work I'm about to tell you about, it wasn't something that we actually set out to study. It was something that to which human genetics led us. We actually didn't go looking for sex-biased genetics. We had really, based on our experiences and that of our colleagues in human genetics, found that in general, almost all of the genetic effects that people have been finding for brain illnesses, and indeed for many illnesses, are present in both magnitudes. And so it's been, although sex-biased biology must be a thing, given the sex biases in disease instances, it seemed like a very hard thing to approach genetically. There just weren't a lot of genetic results pointing to it. And the work that I'm going to tell you about today led us to this topic in a way that we didn't expect. But our thinking about it is still early, and then our thinking about what these genetic results mean is still early. And I would love to get people's thoughts, because I think there's many interesting questions about where one goes from here, from results like I'm going to share in the next half hour or so. So,
yes.
Okay, great. So as this group has surely thought about and talked about a great deal, there are many diseases that show sex-biased incidence or severity for no obvious reason. There are also many diseases that show it for an obvious reason. It's obviously not what I'm going to talk about today, but for obvious reasons, men don't get ovarian cancer and women don't get prostate cancer. But for very non-obvious reasons, women and men also have very distinct vulnerabilities to illnesses that wouldn't be biased for any obvious reason. One of those is schizophrenia. So schizophrenia has a somewhat higher incidence in males. It's maybe about 1.5 to 1, although it's a stronger bias among young adults, people who develop the illness kind of at the beginning of adulthood. But the really striking thing about schizophrenia that my clinical colleagues always talk about is that males tend to have a much more severe course with schizophrenia. They tend to have much more severe negative symptoms, loss of cognition, that there's less remission among the males. And there's this collection of rheumatic autoimmune diseases that I'm going to talk about today. Lupus, or systemic lupus erythematosus, Sjogren's syndrome, and neuromyelitis optica, all of which have much higher incidence in females. So anyway, in addition to showing the strong sex bias, these are also largely illnesses of adults. That is to say, it's not that there's not pediatric forms, it's just that they're rare and extremely severe and not really the typical presentation. And it's also not that people can't get it later in life, but it's just that this is not like most chronic illnesses where risk just keeps increasing as a function of age. These are really all illnesses in which the peak vulnerability is during the reproductive years, and especially toward incidence in the early parts of those years. So in schizophrenia, it had been known for many years in substantial part, really actually foundational contributions in schizophrenia genetics by Sean Purcell and others in our community here, that the human genome's strongest common influence of schizophrenia resides in the major histocompatibility complex locus. That was shown, actually not in this paper that's referenced here, but in 3 papers in 2008, led by Sean and others. And the fact that this was by far the strongest human common genetic association in schizophrenia was in the MHC locus had led to many interesting theories about potential autoimmune or infectious causes of schizophrenia, including this theory described in this story in the Atlantic magazine, How Your Cat is Making You Crazy, describing a theory that schizophrenia arose from a toxoplasma infection that people get from their cats. Fortunately, the Atlantic has actually dialed up its science game a notch for the pandemic. Anyway, there were many theories about infectious causes and autoimmune causes, but it was hard to find the responsible genes and alleles because the MHC locus has very complex long-range patterns of linkage disequilibrium, probably reflecting selective events that occurred in human ancestors in which specific alleles of HLA genes may have been selected for due to protection during epidemics and pandemics during earlier periods in human history. So it was widely expected or even assumed that HLA alleles would explain this association. But HLA alleles didn't appear to explain it. You had to invoke the combination of many, many different HLA alleles to explain to even kind of begin to model the complex pattern of linkage disequilibrium that was present at the locus. And so this kind of mysterious, intractable human genetics problem was actually taken on by Ashwin Sekar, an MD PhD student in the lab, who actually is now a fellow at the Brigham. Ashwin is a hem-onc fellow doing a research residency there. Many of you guys may know him. So Ashwin took on this really daunting statistical genetics problem. He noticed something at the time, which was that if you took this human genetics data that was kind of came from 20 or so different research cohorts, there were specific cohorts in which there was a particularly strong signal, in which the associations were particularly strong to genetic markers in a particular part of the MHC locus. And we actually didn't realize at the time what it was that these cohorts had in common, but they seemed to be pointing toward a specific part of the MHC locus. We now realize, due to the work that I'm going to tell you about today, that these actually were cohorts that were particularly enriched for severe patients and particularly enriched for male patients, just by virtue of the ways that the cohorts had been ascertained, and that we think that's why they had this signal that was really seemed to be pointing to specific markers in this part of the MHC locus. But this caught Ashwin's attention because in this part of the MHC locus, there were these genes, the complement component 4 genes, that had this really complex form of genome variation in which they tended to be present in many different structural forms in different people. And there's actually 2 flavors of the C4 genes. So the C4 genes are highly conserved. They're shared with our rodent ancestors and even back earlier than that in human evolution. But somehow in primates, the ancestral C4 gene duplicated and diverged. And primates now have 2 C4 genes, C4a and C4b. And they only have a small number of amino acid differences, but those amino acid differences are clustered together in a very specific part of the gene that determines what molecular targets are bound by C4 in tissues. So C4a and C4b, despite being almost identical throughout both their coding and non-coding sequences, have maintained for long evolutionary periods of time these amino acid differences that have been shown to be biochemically significant in the sense that they cause the encoded proteins, C4a and C4b, to bind to different molecular targets in tissues. And Ashwin became interested in the question of, like, well, what are all of the alleles and how do they shape human phenotypes? And could they cause these kind of mysterious associations to nearby SNPs? So he developed a set of molecular assays to detect these different C4 gene types. There's both C4a and C4b. And in addition, they exist in both a long and a short form that are distinguished by the insertion of this retrovirus here. He developed molecular assays for precisely measuring the copy number of all these different sequence features. He did these assays in DNA from families so that he could use inheritance to deconvolute these measurements, which are measurements in diploid genomes, into the measurements of transmitted and non-transmitted alleles. And from that, he built models of what are all of the different structural forms of this locus that were segregating in human populations and at what frequencies. And he found that there are 4 or 5 kind of common forms of the C4a, C4b locus that are distinguished by having different numbers of C4a and C4b genes. And that there's a long tail of low frequency and rare alleles. Actually, by now we've found 50 or 60 of them that are also segregating in human populations at much lower frequencies. But most of the variation comes from these kind of 5 or so most common alleles that I'll focus on today. So by measuring the expression levels of C4a and C4b in brain tissue from many people, he found that the expression levels of C4a and C4b kind of scale linearly with the number of copies that people have. So the more C4a genes you have, the more expression of C4a in the brain. The more C4b genes you have, the more expression of C4b in the brain. And then there's this retroviral insertion that distinguishes these genomically longer versions of the gene from the genomically shorter versions. The insertion is completely in an intron, so it doesn't change the coding sequence of the gene. But Ashwin showed that it does change the relative, it does affect the relative expression levels of C4a and C4b. And interestingly, it only does this in the brain. It doesn't do it in immune cells. And that actually is going to be consequential for what I'm going to talk about in terms of a difference between this genetic association in schizophrenia and lupus. So there's all these different alleles or structural forms of the locus. And depending on what combination of alleles you inherit from your parents, you can either have a small amount or a medium amount or a large amount or anywhere in between of C4a expression. And similarly, you can have different amounts of C4b expression. And the other thing that Ashwin showed was that these different structural forms of the locus are segregating on specific SNP haplotypes. That is to say, if you know what combination of alleles a person has for the SNPs that are along this part of the chromosome, that you can actually tell from that what C4 alleles they have. And the reason that you can tell is that the unit of inheritance is a haplotype. These C4 alleles arose on specific ancestral haplotypes in ancestors a very long time ago, thousands of generations ago, and have been transmitted without recombination in between. And so this is actually just significant methodologically, because it means that even if we only have SNP data on someone, we can actually tell, not with 100% certainty, but with fairly high certainty, what C4 alleles they have inherited from their parents. And in fact, he showed that even if you only have this SNP data, that from that you can infer or impute what C4 structure peoples have with a fairly high degree of accuracy. And so this essentially allowed him to turn a molecular biology problem into a big data problem, and to use these large data sets that had been collected by the Psychiatric Genomics Consortium, or aggregated by the Psychiatric Genomics Consortium, genome-wide SNP data from 40 cohorts in 34 countries, to then impute C4 structural alleles into each genome, and then from that, calculate genetically predicted expression levels for C4a and C4b in the brain. And I'm just gonna, in the interest of time, I'm just gonna skip to the punchline that you have this allelic series, it's C4, and these alleles he showed each associates with a different quantitative level of schizophrenia risk. And this allelic series of levels of risk corresponds to the effect that each of these alleles has on the levels of C4a expression in the brain. Interestingly, C4b expression doesn't associate with schizophrenia risk. But there's this correspondence across this allelic series in these relationships. And finally, as I mentioned, each of these C4 structures has actually arisen several different times due to recurrent mutation on different MHC SNP haplotypes. And Ashwin showed that regardless of which haplotype a C4 structure has arisen on, it tends to associate with the same level of schizophrenia risk. And again, that this corresponds to the level of C4a expression that allele generates in the brain. And finally, Ashwin showed, but this has actually now been replicated in two other panels of postmortem brain tissue from different cohorts, that expression of C4a is elevated in brain tissue from schizophrenia patients relative to its level in controls. And interestingly, the magnitude of this effect is several times greater than you would predict just from the fact that people with schizophrenia tend to have high C4a alleles. That is to say, the data suggests that above and beyond the contribution of at the C4 that schizophrenia patients tend to have higher levels of C4 expression in their brain to an extent that can't be explained by genetics, and that actually has gotten us really interested in and actually a whole group of neuroscientists interested in C4 as a potential biomarker for dynamic changes. That is, C4 measurements, C4 protein measurements in CSF is a potential marker for dynamic changes that are relevant to the course of schizophrenia. Finally, experiments by Heather de Rivera in my lab showed that C4 protein is actually present at synapses. Of course, the question, of course, is what is it actually doing at synapses? There's, as you're probably aware, it's been known for the complement system, and in particular, this whole cascade of complement proteins has been known for many years for its role in the innate immune system, in which this cascade reaction by complement proteins tags cells and debris for engulfment by macrophages. And in particular, a molecular debris that's being targeted for degradation becomes decorated with C4. That then instructs the deposition of another complement protein, C3, onto the same molecular structures. And then C3 in turn interacts with a C3 receptor that's expressed on phagocytic cells such as macrophages, and causes these structures to be phagocytosed by macrophages. There's really interesting work by Beth Stevens's lab, and before that, when Beth was a postdoc in Ben Barris's lab, that's found analogous, in some ways kind of functionally analogous roles for this molecular system in the brain in tagging synapses for engulfment by microglia. You can see how in some ways there's a nice analogy in the sense that it's a set of proteins being used to decorate molecular structures and target them for phagocytosis by immune cells of the myeloid lineage in the brain by microglia and in the periphery by macrophages. And as part of the work that we published on this schizophrenia result, a postdoc in Beth Stevens and Mike Carroll's lab showed that C4 also instructs this process of tagging synapses and recruiting C3 to synapses, which is a signal for their engulfment by microglia. I'm not going to talk more about that work today. And in fact, we've actually kind of tried to stay as agnostic as we can to what the cellular and molecular mechanism of this human genetic result is until it's kind of been shown, beyond a shadow of a doubt, which I don't think it has at this point. So I think that this may well be the mechanism for the association of C4 with schizophrenia, but we don't know that yet. And that's the subject of ongoing work in Beth's lab. So I should say the mechanism of pruning is a plausible connection between this genetic discovery and earlier observations that schizophrenia patients have tend to have fewer dendritic spines on certain neurons in the cerebral cortex. But it is still possible. Complement may affect brain function and synaptic function via mechanisms other than synaptic pruning. And although I'm not going to talk about it as much today, we've really come to, maybe more in the discussion, we've really come to think that even if C4 effects are due to synaptic loss, that this may be primarily affecting illness severity rather than being the primary or initiating pathology in the illness. But this actually just leads me to, I want to talk next about C4 in autoimmune disease. So there was actually something really interesting that was noticed by our colleague, Mark Daly, which is that there's this inverse relationship between schizophrenia risk alleles and their effect size. That is to say that the more common allele is, the smaller its effect size tends to be on schizophrenia risk. And the interpretation of this is just that there's probably in human ancestors been selection against common alleles that shape risk of schizophrenia. The strong exception to this pattern are the alleles at the C4 locus, which just have again, they're not acting like Mendelian alleles, but they still have a stronger effect size than one would expect based on their allele frequency. And this had often caused us to wonder whether in a different biological context, these same C4 alleles that elevate risk for schizophrenia might be protective in another context. And after kind of exploring C4 effects in actually many illnesses and finding that in general C4 was, even in many illnesses with MHC associations, that these were not due to C4, we actually found an illness in which we became actually quite certain that the strongest genetic effects actually really are due to C4, due to the research that I'm going to tell you about in the next few slides. So lupus is a systemic autoimmune disease in which the immune system attacks healthy tissue in many parts of the body. The usual onset is similar, as in schizophrenia, the usual onset is around 15 to 45 years of age. There's no cure. It's treated with steroids and other immune suppressants. The cause of it is unknown, and it's far more common among women of childbearing age than among men of the same age. It's about 9 times more common. There's been really good human genetics that's been done on lupus, led by Lindsay Criswell and Rob Graham and Tim Weiss and many others, all of whom we collaborated with in the work I'm going to tell you about. The strongest association in lupus by far is to the major histocompatibility complex locus. And it has actually been believed, this was discovered actually even long before the GWAS era, just from, because you can type HLA markers molecularly on cells long before you could interrogate people's genomes with SNP arrays. And so it's actually been believed for decades that this strong genetic association is due to variation in the HLA genes. And in part is believed that because that is true for most autoimmune diseases. But there's a really important difference. In many of these other autoimmune diseases with strong HLA associations, such as MS and type 1 diabetes, the implicated variation has been shown to affect HLA genes, to affect specific peptide binding grooves and to determine which antigens can be presented. And so, for example, in type 1 diabetes, for example, there's an HLA allele that's almost strictly required for the disease to develop and that actually presents an autoantigen that's critical in the disease. But lupus is very different. In lupus, there was never a clear association of specific HLA alleles with specific autoantibodies. And the observed genetic signal is complex. As in schizophrenia, it requires many different HLA alleles to explain. So we wondered whether the HLA alleles were actually really genetic markers for something else. And this is work is led by Nolan Kamatake. He actually started this work as an undergraduate in the lab and then came to the lab full time and has finished it. And this is the work that was just published in Nature a few weeks ago. So Nolan and a colleague in the lab, Bob Hansiker, developed ways to expand this, this panel of reference haplotypes for C4 so that we could infer or impute even more C4 alleles. And so in some of the analyses that I'm going to show you, there's now, the analyses are shaped by a improved ability to impute these C4 alleles and to impute a larger number of them, including several of the lower frequency alleles that I alluded to before. And so just to make a long story short, by imputing these C4 alleles into these large human genetics cohorts that had been collected for genome-wide association studies in lupus, Nolan found this really striking result. And this is sort of an unconventional way of showing, of reporting human genetic results. So I'm just gonna dawdle on it for a few minutes to just explain, because there's actually several genetic associations that are represented here. So here, in this plot, each of these circles represents some combination of like the number of C4a genes someone has and the number of C4b genes that they have. And the area of the circle is proportional to how many people have that combination of C4a genes and C4b genes in their genomes. And then Nolan has colored each of these circles by the frequency of lupus in that group of people. So it's basically colored by a kind of an odds ratio that shows the relative risk for lupus of these different groups of people. And the large group in the middle is the most common combination of C4a and C4b copy numbers to have. And that's kind of shown here as a kind of a neutral risk. And then but what you can see, and actually, this is actually on a log odds ratio. We probably shouldn't have done this because it's sort of in this, I think we did it differently in the paper, but this sort of understates the magnitude of risk. That there's roughly, this group of people is at roughly 8 times the risk of this group of people. And what Nolan found is that both C4a and C4b protect for lupus. So you can see as you kind of travel from left to right on the plot, that if you control for C4b gene copy number, that the more C4a genes people have, the less their risk of lupus. But actually, if you, C4b in lupus appears to also be protective. So you can see as you make kind of vertical paths up the plot, that as you increase the number of C4b gene copies, that is also protective, although less, it's less than half as protective per gene copy as C4a is. And finally, you can see from diagonal paths the effect of exchanging C4a for C4b, which is that's also protective. So they're both protective, but C4a is more protective than C4b is. And these effects are similar. We were fortunate that there were actually large cohorts of African-American research participants on which we could also draw. And all of these effects for these different C4 alleles appear to be indistinguishable effect size in Europeans and African Americans. But African American cohorts were, in analyses I'm not going to show today, really critical for showing that that this genetic effect was not due to the nearby HLA alleles. Because in Europeans, due to linkage disequilibrium, it was really hard to separate the effects of HLA alleles from C4 alleles, but Nolan's analyses in African American cohorts made this distinction very clear. And it is actually something that, this is a really important gift that African American research participants have given to this area of human genetics, which was that it was really thought for decades that this genetic association was due to HLA gene. And it becomes really clear when you look in African American research cohorts that it's not. Sjogren's syndrome is an illness that has many things in common with lupus. It's also an autoimmune disorder, but it tends to be one in which the exocrine glands are particularly strongly affected. It's characterized by dry eyes and mouth, but as in lupus, with kind of other systemic effects. And like lupus, it's also characterized by diverse autoantibodies, not antibodies to like one disease-critical autoantigen, but antibodies to a wide array of antigens, including anti-nuclear antibodies that target ribonuclear proteins. And as in lupus, it's the strongest common genetic risk factor that had been found is also in the MHC locus, with associations to the same SNPs and haplotypes as in lupus. And here Nolan found, when he reanalyzed this data in much the same ways that he had the lupus genetics data, that it's really a very similar story as in lupus, that the same alleles are high risk in both disorders, and in fact, actually, the effect size is much greater in Sjogren's than it is in lupus, which is fortunate because it's a somewhat less common illness. So it actually would have been hard to see these effects as clearly in the smaller research cohorts that are available for Sjogren's. But anyway, this particular allele, which has no copies of C4a, and is actually the most protective allele in schizophrenia, is the highest risk allele for both of these autoimmune disorders. And similarly across the whole allelic series. And neuromyelitis optica is another autoimmune disease. It's much rarer, and so this analysis is much less satisfying, and the confidence intervals are wider. But we've come to believe that C4 is also similarly the reason that neuromyelitis optica associates with the MHC locus. So how might complement be protecting against these illnesses? So this is what we think is going on. So in the context of there's cell damage in our bodies all the time, and just as parts of natural processes, but also all kinds of things, including actually things like sunburns, pregnancy. All of these things can cause cell damage in various parts of the body. And whenever cell damage occurs, it exposes the immune system to nuclear debris, which it wouldn't normally see, in the context of cell injury and distress signals. It's very much the kinds of distress signals that cause the adaptive immune system to go look at antigens. And what we think is happening is that in these contexts, there's the potential to break tolerance, leading to autoimmunity against ribonuclear proteins that the adaptive immune system isn't normally, and in particular, is the antibody-based adaptive immune system isn't normally seeing. Complement proteins have been shown to bind to ribonuclear proteins incredibly quickly, facilitating their clearance and attenuating their exposure to the adaptive immune system. So what we think may be happening is that the more, and I should say in work from our colleague McCarroll's lab that is under review, but that he's given me permission to mention, they've shown that C4a does this more effectively and more quickly than C4b does. That is to say that they both help to clear ribonuclear proteins, but that C4a magnifies the speed of this process. And that, so what we think is happening is that these complement proteins are attenuating the visibility of nuclear antigens to the adaptive immune system. And that may be the mechanism by which having more copies of C4b, or especially C4a, offers people quantitative protection against the development of autoimmunity, particularly in these illnesses in which people develop antibodies against a wide variety of ribonuclear proteins. So we've talked about complement genes in schizophrenia and complement genes in autoimmune disease, but now I want to actually bring it to the topic that I know that this group is most interested in, which is complement and sex differences. And so it was really striking to us how these C4 alleles, first of all, had opposite effects on the risk of schizophrenia and of these autoimmune diseases. That is to say that the same allele, that having more C4 expression increases risk for schizophrenia, but is offering strong protection from this constellation of autoimmune illnesses. And so we naturally wondered whether, and as I mentioned before, these illnesses have very different sex biases, that schizophrenia tends to be much more severe in male patients, and that women are much more likely to be affected by lupus and Sjogren's and neuromyelitis optica. And so that just made us, that made us wonder whether these C4 alleles, which appeared to be the strongest common genetic effect in all of these illnesses, might be acting in a different way in males and females in these illnesses, and in particular, whether they might be acting more strongly in males. And this turned out to be the case in both lupus and schizophrenia. I should say in Sjogren's, the data kind of points in the same direction, but it isn't as clear just because the cohorts are not as large. It's a less common illness than lupus. But in both lupus and schizophrenia, each of these alleles seems to act with a larger effect size in men than in women. The genetic effects are clearly present in both men and women, and the same alleles that are strongest in men are strongest in women, and vice versa. But not only are the effects in opposite directions in the two illnesses, but in both illnesses, those effects are stronger in men than in women. This is just another way of presenting the same data across the allelic series in terms of odds ratios, but just showing that in men that, I mislabeled this slide. It should be schizophrenia on the left and lupus on the right, so I'm just gonna skip it. So why might C4 alleles shape risk more strongly in men? Possibility might be that the greater levels of, might be that the genes, each copy of the genes is transcribed into more RNA in men than in women. Another possibility might be that RNA is translated into mature protein and prepared for secretion and secreted more in men than in women. And then the third possibility, that would be most complex and hardest to address, would be that there might be differences in downstream responses to complement. So this first possibility was something that we were very quickly able to rule out, because the GTEx project has profiled RNA expression in hundreds of men and women in scores of different human tissues. And for the most part, the C4 genes are at fairly indistinguishable levels of expression in men and women. But we found that C4 is present as a protein at very different levels in the cerebral spinal fluid of men and women. And again, this plot is actually on a log scale, so it may understate the differences a little bit, but men on average have substantially higher levels of C4 protein than women do. And in fact, in the CSF. And in fact, this appears to also be true of C3. And I should say that, these experiments that we did found this to be true in CSF. This was already known to be, this was already well established in plasma. in which it is just collected on far larger scales than cerebral spinal fluid. That is to say that the levels seem to be higher in men than in women. And this is true across ages. So here, this is kind of the same analysis, but just broken down by people's ages. And you can see that, yes, there's a substantial amount of inter-individual variation, but the overall pattern tends to be that on average, men have higher levels than women, but this is primarily true during the reproductive years and not before or after that. And the same thing for C3. So just to summarize, alleles of C4 that contribute to schizophrenia risk appear to be protective for multiple autoimmune diseases. The C4 alleles appear to affect risk in both sexes, but to do so more strongly in males. And that these C4 genetic effects may act multiplicatively with sex differences in the tendency of C4 genes to produce mature secreted protein in males and females. And so finally, I'd like to thank the people who contributed to this work, especially the two students who really just did amazing work taking what were considered kind of intractable human genetics problems and wrestling them to the ground, as well as the way this work has benefited from collaboration and conversations and ideas with many people in our research community, especially McCarroll and Beth Stevens. So thank you very much. I'm really sorry for the AV difficulties, but I would love in any time that we still have to take any questions that people might have.
Great. Well, thank you so much, Steve. Thanks for a fascinating talk. Do people have questions? You can either unmute yourself and ask them, or you can type them up in the chat window. Maybe while you're thinking, I'll ask a first question. What cell types do you think C4 is being expressed in, where it's exerting its effects in schizophrenia and in autoimmune diseases? Are they the same cell types or different?
Definitely different cell types in the two illnesses. So in schizophrenia, C4, it's expressed in neurons, but it's expressed more highly in astrocytes and oligodendrocytes. But it's really hard to say. I don't think one wants to say just based on a quantitatively higher expression level, which it is. But we know that it's expressed in neurons, and we know that in neurons, it can get to synapses in a neuron autonomous manner. So it doesn't need microglia or glia for it to get to synapses. But we don't know for sure which is the important cell type kind of driving those effects. In the periphery, C4 and C3 are expressed very strongly in the liver. And in fact, the liver makes a lot of the circulating complement proteins that are circulating around the body. But it also is expressed, for example, in the lymph nodes and stromal cells and things like that. So there's multiple places where it could be contributing to this biology. It may just be that it's the secreted protein that's flowing around the plasma and around the CSF that's then depositing on these molecular structures. That would be my guess, but we don't know that for sure.
Other questions?
So I'll ask a question. This might be a simplistic question, given the complexity of the immune system, and I'm in no way an immunologist, but you kind of talked about the genetic risk associated, particularly interested in the schizophrenia genetic risk, but you alluded to the idea that it seems to be also closely linked with severity of illness. I'm wondering if there's any evidence that sort of C4 expression or the levels of C4 either in CSF or in the periphery are at all associated with potentially looking at a consequence of disease as opposed to risk for disease, looking at exacerbations of symptoms and immune activation and that kind of downstream effect.
Yeah, so this is an idea that we've really gotten interested in the context of schizophrenia. And one of the things that got us interested in this idea actually is this result, because it just really seemed like whenever we talk with people who work with clinicians, the thing that they're really struck by as the sex difference in schizophrenia is severity. And that has really led us toward thinking that the C4 genes may actually be contributing not in the kind of initial stages of the illness, but potentially in the kind of progressive period of the illness in which people's cognitive symptoms get worse and worse. And I know that can at first seem puzzling because obviously we discovered that genetic effect in a case control study that was just comparing frequency of alleles, cases, and controls. So on some level, you tend to see us to think like it wasn't meant to be a severity study, but there's a sense in which severity can, the sicker someone is, the more often they're going to be going to the hospital, and the more likely they are to get ascertained and do a genetic study. And especially, I mentioned that some of these genetic effects were strongest in certain cohorts that had ascertained the sickest patients, that had done like kind of hospital ascertainment as opposed to inpatient ascertainment. As opposed to outpatient ascertainment, for example. And so the more often someone's going to the hospital with severe symptoms, the more likely they are to be ascertained. So I think severity can end up, even when that's not part of the study design. And in fact, even in that Swedish cohort, the initial phases of it were focused on, patient collection were focused on the most severely ill patients. And then with time, the ascertainment became broader, and this genetic effect actually weakened. And so we're thinking more and more that this may be a severity locus. There's one result that we have in that direction, is not published yet, but when we measure complement proteins in the CSF of patients who've been ill for many years, that confirms this result in postmortem brain, that C4 is elevated in cases relative to controls. But when we look at first episode patients, we really don't see that clear elevation at that point in the illness. And so it's small numbers yet, and that's why we haven't published it yet, but we feel like the data is kind of pointing in this direction of it potentially being part of the progressive escalating period of the illness, as opposed to being maybe like the initial vulnerability to the illness. Now, it doesn't mean that it's that way in lupus. It may be actually quite different in lupus. And in fact, in lupus, there's evidence that once you have established, that complement can make the pathology worse. So complement can definitely make immune pathology worse. There's actually evidence that that C4 is contributing to in some of the more severe COVID cases, when people have cytokine storms and things like that. There's definitely a lot of complement deposition also. So it's possible that C4 is contributing to some of the sex differences in there, where men seeming to be enriched among the patients who do the worst with COVID. And so, yeah, that aspect may be quite different in different illnesses.
Thanks. We have a question from Brittany Bauman in the chat. Brittany, do you want to unmute yourself to ask the question?
Hi. So I was just curious, so you had shown a slide with differences in men and women. I can't recall exactly what you were showing. It was the blue and pink slide. Oh
yeah, so two slides actually. So one. Yeah. So one is just in the sex differences in vulnerability, and that men get schizophrenia more often than men get schizophrenia more often than women do, but that really only applies to young adult onset schizophrenia. There's actually a later phase at which people some people actually develop schizophrenia later in life, for example, in their 40s, but that is not more common in men. In fact, if anything, that's more common in women. So it's more, and the same thing in lupus and Sjogren's, that these are adult onset illnesses, and that the difference between male and female onset is really among patients who develop the illness in their reproductive years and not among people who develop the illness earlier or later. And then that also seems to be true for levels of circulating C4 and C3 in both the plasma and the CSF, that these sex differences are present during the reproductive years, but they're not present before or after that.
So I guess adding on to that, have you made any hypotheses in terms of like endocrine differences into why that is, like that specific reproductive age onset, or like protective effects of certain hormones, or I don't know, just going on?
Yes, so we've had a variety of hypotheses about it, but these things are actually hard to show.
Sure.
You know, they're really hard to show because as you know, these endocrines, they're complicated systems. These are things that are kind of hard to show in like cell culture, right? You in many ways probably need, because these are complex systems with many organs sending signals to each other and respond to each other and things like that. And so, some basic kind of cell culture experiments, we found that, we didn't find that testosterone or estrogen elicited differences in the maturation of C4 in cultured cells. But that doesn't mean that that wouldn't necessarily be true in a whole person or a whole animal, which is obviously a very different system. I should say our colleague McCarroll, his lab has now confirmed in mice that this difference between males and females in levels of circulating complement proteins they do see that in mice. So in principle, that aspect of the biology may be possible to study in mice. It seems to be the case that although males and females transcribe these genes at the same rate, that there's something in the male system that causes more protein to be secreted.
All right, thank you.
But we really think it's happening at that level, not at the level of transcriptional regulation, but at the level of protein regulation and particularly potentially of the maturation and secretion of these proteins, which go through many proteolytic steps on their way to being secreted. And there's some evidence that some of the proteins involved in those steps can be regulated or can be regulated by sex hormones. But that's not our work. That's earlier work.
Is there any evidence that any of those processing genes are genetically linked to schizophrenia? I mean, it's very highly heritable, and as I understand, the C4 counts only for a small fraction of the genetic risk.
Yes. So actually, this is kind of more unpublished work, not even enough really to put in a slide, but we found some evidence that that the furin protease is one of the so I should say furin is definitely a protein that contributes to the maturation of C3 and C4, and common variants in schizophrenia, common variants at the furin locus definitely associate with both schizophrenia and lupus. But I think it's just gonna take some more work to sort of establish kind of clear cause and effect and see how that's being regulated by sex hormones, or whether that, sex hormones almost certainly do more than one thing, right? So that you can fall into the trap of like, because you've seen one thing in the lab, you think that must be the thing. And so we're kind of trying to be careful how we proceed, but we think that might be one of the levers of regulation.
Very interesting. Other questions?
Hi, Mike. Sean here. Great talk. Is it fair to say that the C4 association here is exceptional, perhaps in at least two ways, right? So one, as you pointed out, in terms of the effect size and the frequency of the association, but also perhaps in its not sex specificity, but the sex difference you alluded to here. In as much as, and actually I have to say I'm not completely up to date with the latest, but in general, isn't it fair to say, the genetic correlation between men and women is basically unity, heritabilities are similar, and so on, and sex stratified analyses don't obviously yield different results in most cases, right? Say in schizophrenia, GWAS. And so if you agree with that, I'm just wondering, are those two things connected? You know, for example, one can imagine just how sex-specific expression is going to impact different, have an impact on selective pressures on variants and so on. In other words, it's perhaps because of sex specificity, it allows for a larger effect in this way. So just the connection between those two aspects of the association and I guess more generally, what this tells you or doesn't tell you about how to think about sex-specific biology and genetic effects beyond C4 in schizophrenia, if that's clear.
Yeah. So I completely agree. There's been very little in the human genetics of these complex disorders that is strongly sex biased. That it really, in general, if you look at genetic, even for sex biased disorders, in general, if you look at genetic effects, they kind of have the same effect size in men and women, and the exceptions tend to be for kind of highly. I think there's some exceptions for things like waist-hip ratio and things like that. But for traits for which there's like obvious sex dimorphism, right? And so it's a really interesting question that you point out, whether the large effect size is somehow connected to the fact that it also varies between the sexes. There's something about this biology that somehow, that somehow has been, I mean, I guess just in the fact that it can vary in this kind of, in this sexually modulated way, just has given, somehow the biology of this system has allowed for variation, both allelic variation and sex differences, to intrude in substantial ways, in ways that a lot of other aspects of biology don't. And I don't know what it is about the complement system that has accommodated that. It may well be that, I think they're almost certainly due to, so certainly pregnancy is a very major physical event that the female but not the male body undergoes, and it's both an event that actually can cause quite a bit of cell damage, and in fact lupus sometimes there's an enrichment of lupus manifestation actually in the years after childbirth, after one's first childbirth, but also carrying a fetus with a different genome creates an immune context that also is very singular, and in which that's a very kind of immune special space in terms of how the adaptive immune system is supposed to behave there. And so that may also have been kind of what created part of the latitude for this system to vary more. And it also may be that the ways in which it varied kind of had to be calibrated well. It had to be recalibrated at different times in evolutionary history as we faced different kinds of immune challenges. So it may just be a part of our biology that's just been very active. I mean, that's been changing in recently in human evolution.
Got it. Thank you.
Great. Thank you. Any last question before we wrap up? If not, as they say, going, gone. So thank you so much, Steve. Fantastic talk, absolutely fascinating material. And thank you to everybody for your patience through the technical glitches. And we thank Steve again. And just to remind you that we have another talk on September the 30th. Eric Nestler is our next speaker. So that's Wednesday, September the 30th. And hope to see many of you there. So thank you again, everybody, and have a great evening.